here at JPM, and I'm here with Angelos Stergiou. He's the CEO and founder of Sellas Life Sciences. And yeah, just first of all, give us a bit of context about who you guys are and what you're doing here.
Absolutely. Thank you, and great connecting with you, Max. So Sellas Life Sciences, we are an immuno-oncology company based in New York, and obviously focused predominantly in the oncology and the immunotherapy space, and in hematological as well as solid cancers. We have two programs, two assets. One is galinpepimut-S, or GPS. It's a technology that we licensed in out of Memorial Sloan Kettering, who over the years have been great partners and collaborators. It's a technology that David Scheinberg, out of his lab when he was the chief of leukemia at Memorial Sloan Kettering, had looked at, because in leukemia over the years, no real advancement had been made. And so he really was looking for something cutting edge, novel. So when we licensed the program in a few years back, we had phase 1 and 2 data. But what's really neat about this asset in particular is that we target Wilms tumor 1. The NCI actually ranked it as a number 1 immunotherapy target to go after a priori selected criteria. In the fetal state, Wilms tumor 1, so it's a fetal oncoprotein. It resides in the nucleus, but in the fetal state, it's responsible, among others, for kidney formation. But once we're born, it disappears, comes back again when it causes cancer. So it's overexpressed in about 20 tumor types. In leukemia, 97% of patients express WT1. Mm-hmm. So it's really a great technology. Again, for leukemia, mesothelioma, everybody expresses WT1. Then you have other indications or tumors, like ovarian cancer, where around 30% or so of patients express WT1. So there, you really want to do like a PCR testing, which is very easy to treat the appropriate patient population. So the technology, again, out of Memorial Sloan Kettering, and Memorial Sloan Kettering, David Scheinberg, back then had used sort of artificial intelligence really in the earlier days, mm-hmm. And came up with 4 peptide sequences. We target 25 carefully selected WT1 epitopes. Importantly, we've utilized something called the heteroclitic technology. It's by design mutation of amino acid sequences, and what that allows us to do is, it breaks tolerance, so we can administer our GPS drug really ad infinitum. And secondly, it makes it really more potent. And again, with the introduction of the tetraclitic technology concept, and through MHC class 1 and 2 presentations, we elicit both CD4 and CD8 immune response, which is really important in this space, because old technology, they would only elicit a short-lived cytotoxic CD8 T-cell response. And that's just not enough. You really need those CD4 helper and memory cells to really go out and really support the immune system. So what we do with our treatments is subcutaneous injection. We provide the picture to the immune system, to your body, what the cancerous cells look like that carry, if you will, the WT1. And if and when the cancer comes back, if a patient was in remission, for instance, it laser focusly finds those cancer cells and kills them. So that allows for patients to stay in longer progression-free survival, and ultimately, and most importantly, to prolong overall survival. Amazing. So this is the one asset, and I'll talk about the clinical data in a second, but sort of about GPS. And then the other program that we have, to talk a little bit about the science first, and again, if it's okay, Max, we can then talk about the clinical data. Definitely. It's a CDK9 inhibitor. It's a highly selective CDK9 inhibitor, and we had licensed in the program about 3 years ago. And we were very bullish in our statements when we said that we are and we have the most selective CDK9 inhibitor. And we said that obviously on scientific merits. We had done a lot of work and diligence when we brought it in. What's interesting is that many cancer cells, they're sort of, if you will, they need fuel to stay alive and or to not get killed. And you have certain proteins like MCL1 and MEG, and they depend on that CDK9 pathway, if you will. So what we do is we block CDK9, and basically as a result we can kill cancerous cells that again depend on the CDK9, and so there's much more biology behind it, but that's sort of on high level. But I think what's differentiating us to other CDK9 inhibitors is, again, we have done work, and CDK9 belongs to a bigger CDK family. But when we looked through all the kinome proteins and compared our CDK9 inhibitor to other competitors out there, we saw that, and again, we looked at the kinome proteins, that ours is really selective to CDK9, versus you would see other CDK9 inhibitors that were sort of all over the place, and as a result would have off-target toxicity. So we are in the body long enough to kill cancerous cells, but we're not in the body long enough to cause severe unwanted toxicities. And I think that's, so from a pharmacokinetic perspective, that's one differentiator. And obviously safety is crucially important. At the same time, efficacy is a key driver, and we have seen really exciting data so far.
Yeah, that's amazing. Yeah, let's just go on to what last year brought you, but also you've got a big year ahead. Let's talk us through what 2025 will bring.
Yeah, we have a really exciting year ahead, and to your point, with our first asset, GPS, that I alluded to before, of Memorial Sloan Kettering, in previous phase 2 work, among others. So we had studied GPS in various indications, both in hematological malignancies, in leukemia and myeloma, as well as in solid cancers, including ovarian cancer and mesothelioma. But in leukemia specifically, if I may, we've done work in patients after first complete remission as well as second complete remission. In the former, in patients after first complete remission, and by the way, the majority of AML patients are ordinarily about 60 years and older. Mm-hmm. In patients after first complete remission, in those patients, we had seen a 35.5-month median overall survival, which was about double or triple of what you would expect versus historical data. But then in the second remission, so patients after second complete remission, we showed a 21-month versus 5.4-month median survival benefit in favor of GPS with a p-value of 0.02. That allowed us then to move into a phase 3 program. Wow. So we have enrolled 126 patients, in patients in CR2, 1-to-1 randomized. And in December of 24, we triggered the 60th event, and that with event meaning deaths, which triggered the interim analysis. And this month, in January of 25, the Independent Data Monitoring Committee will convene, and there are really 3 potential outcomes. One is they may stop the study for futility. Mm-hmm. It means that GPS just didn't benefit the patients. The second thing that could happen is that the IDMC will recommend to stop the study because of already superb efficacy. And the third outcome is to continue for another 20 events, so for another 20 deaths. Again, the interim is at 60. And to then basically see what the data readout will be. We as a company, we're hoping that the study will stop for early efficacy now, although the bar is very high, but we're very bullish with our asset. But at the same time, we would be equally excited if we have to go for 20 more events. Why? Because that also means that we have passed the futility testing, because otherwise we wouldn't be continuing to the final study readout. But we would just have to wait a little bit longer. So certainly, what we are hopeful for and very excited is that patients first and foremost will benefit from GPS, because next to the efficacy profile, important to note that it's really safe.
Yeah.
The safety profile is superb, if I may say. It's very similar to a flu shot or a COVID vaccine. Those are sort of the side effects. So on GPS Max, milestone-wise, in January, IDMC meeting, and we hope for good news, obviously, both for patients and our shareholders alike. The second program, CDK9, our SLS009 program.
Mm-hmm.
In 24, we are obviously focused in patients, relapse refractory AML patients, and those are relapse refractory to venetoclax-based treatment regimens. Why? Because certainly in the US and in Europe, there's just about no patient now who's been diagnosed with AML who has not been introduced venetoclax, and it's one of those backbone treatments in AML. The problem is, though, that the majority, if not all patients, at some point will relapse,
Okay.
or they're refractory to begin with. And refractory we define as at least 3 cycles of treatment, and again, the drug just doesn't seem to work. What we had seen preclinically, and we've validated in the clinic, is that patients who are in venetoclax and we add on our drug, we can make that response even stronger. Or those patients where venetoclax didn't work to begin with, we can basically sensitize them and get them to respond. And again, there's a lot of biology behind that. The other thing that we did in 24, and I'll tell you in a second what to look out for in 25, we identified ASXL1 mutation. That ASXL1 mutation is found both in hematological malignancies, like in AML,
Mm-hmm.
as well as in CML and other indications, including in solid cancers, predominantly in colorectal cancer and non-small cell lung cancer and neuroendocrine tumor types. So with that ASXL1 mutation and our clinical team, jointly with institutions out at MD Anderson, North Carolina and Alabama, we were able to really, in a way, establish almost a precision type of modality treatment approach, because we do know that relapse refractory AML patients with that ASXL1 mutation do very well on our treatment. So in the early studies, in the phase 2 program, we've now shown in 30 milligram twice a week administration of our drug that patients' median overall survival has not been reached yet.
Mm-hmm.
But it's 8 months and counting. Those patients ordinarily die within 2.5 to 3 months. So we have already almost shown a 3-fold survival benefit and counting. And we have also seen a response rate that exceeds 50%, and the response rate right now with standard treatment is around 20%. So that's what we call AML MRC patients. Those are mutation-related changes. And there's an array of mutations in those patients. ASXL1 is a key driver. But what we have seen is that with those MRC AML patients, with an ASXL1 mutation or not, benefit from our treatment. Again, there's a lot of biology behind it that I think it's too complicated and will take time to explain. But I think suffice it to say, we're very excited with what we have seen, and we're in discussions with the FDA to see what the regulatory and clinical path will look like, because we obviously want to expeditiously proceed with the program that will ultimately and hopefully go to the market soon to benefit patients. Again, we're sort of focusing with our CDK9 inhibitor in those relapse refractory patients. We're also going to move into frontline with our intention of our CDK9 program, because ultimately, Max, with SLS009, with our CDK9 program, we can treat the AML patients sort of in the state frontline and get them into remission and treat them.
Yeah.
And then GPS is a maintenance therapy, which would then basically come right after that piece is completed. So we can really embrace the entire AML space, if you will. An additional thing from 24, so milestone for CDK9 this calendar year will be the FDA feedback that we expect in the first half of this year, and so we're really excited about that in relapsed refractory AML. But we had also done work in 24 where we showed that in colorectal cancer and in non-small cell lung cancer, our drug was highly efficacious. We are embarking on potential non-dilutive funding to advance CDK9, our SLS009 program in frontline AML, but also to continue some work in solid cancers. And I think as an additional point to also be looking out for this calendar year is some data in pediatric tumor types. And we've done some work there because the NCI awarded us the PIVOT program. So all in all, yes, it's a very exciting year for us here at Sellas 2025. Very excited, what this year will hold for the company, but most importantly for the patients.
Yeah, it's amazing. It's amazing. So much to happen. So much excitement. Best of luck with the rest of this week.
Thank you. It's been a busy week, yeah.
And amazing to have you. Thank you.
Thank you. Thanks, Max. Thank you.